Okay, so we have reached today the end of immunoglobulin gene organization. Okay, we'll start with the class switching among the constant region genes. So class switching among the constant region genes, that is heavy chain, constant region genes. Okay, so the class switching you have studied in B cell activation, how the somatic permutation and the class switching takes place. So here the VH, DH and JH, that is the heavy chain, variable D and J region, the unit combined with these units combine with any of the CH gene segment, okay, any of the constant gene segment, which is also known as the isotype switching. So VH, heavy chain variable region, so that can combine with any of the constant gene segment, which is known as isotype switching. This takes place after the antigenic stimulation of the B cell. So this involves the class switching involves the DNA flanking sequences called as switch regions, okay, which consists of 2 to 10 kb. So switch regions composed of short repeats, that is GA, GCT, TG, GGG, like this. The short repeats are present in the switch regions. So cytokines, the cytokines you know cytokines, they are the intracellular regulatory proteins. They act as a switch factors. Cytokines act as a switch factors, plays a major role in determining the immunoglobulin class expressed as a result of switching. So which class of the immunoglobulin it is, whether it is M, D or E or whatever it is. Okay, so which class is expressed? So the variable region that can combine with any of the CH gene segment, that is known as a isotype switching. That involves the flanking sequences called as the switch regions. So the cytokines, they are known as switch factors. These cytokines plays a major role in determining the immunoglobulin class, which is expressed as a result of the switching. So which class is expressed? So here you can see this is the proposed mechanism for class switching. So you need not learn this, just for your understanding. Proposed mechanism for class switching induced by interleukin 4, it is a cytokine, in rearranged immunoglobulin heavy chain genes. Okay, so here the switch site is located upstream from the each CH segment. Okay, so except C delta, so this is and this one S mu and S gamma 1. So here the looping takes place, DNA looping takes place. So this is how it forms the loop, and then joining of S mu and this one takes place. Okay, this is the one, S mu and the S mu and the S gamma 1. So joining of these two takes place. Okay, so this is how. So after that, so this removal of this, that is cutting down, and recombination at this region, S mu and S gamma 1. So you will get this one. So again, one more looping takes place from S gamma 1 to S epsilon. So DNA looping and recombination again. So you will end up with these two, that is S mu, S gamma 1, and the other one it is S epsilon. So this is how the immunoglobulin heavy chain genes are rearranged, okay, for class switching. Uh, Next, expression of the immunoglobulin genes. Expression of the rearrangement is done. Next, expression of the immunoglobulin genes. So, expression of the immunoglobulin genes, that means formation of the mRNA, and then it is translated to the immunoglobulin. So, processing of the primary transcript, that means after the gene rearrangement, transcription takes place, that is known as a primary transcript. Primary transcript to remove the intervening sequence including non-coding introns and the gene segments not lost during the VDJ rearrangement. So what is not lost? During the processing of the primary transcript, to remove the intervening sequence that includes the non-coding introns and the gene segments which are not lost during the rearrangement. So that should be processed here. The remaining exons are connected by a process called as RNA splicing process, and then the final mRNA product is then exported to the nucleus and then translated with the ribosomes into complete heavy and the light chains. So processing of the primary transcript, and then the exons are connected by a process called as RNA splicing, and then the final mRNA product is then exported to the nucleus, and where it is translated by ribosomes into complete heavy and the light chains of the immunoglobulin. So that is how they are expressed. Immunoglobulin genes are expressed. Next, differential RNA processing of heavy chain primary transcripts. So you have heavy chain primary transcript. So how differential RNA processing takes place here? So we'll see how it is. Immunoglobulin can exist in either the membrane bound or the secreted form. So membrane bound immunoglobulin or secreted immunoglobulin. So the processing can yield different mRNAs. During processing, for the membrane bound and for the secretory, the processing is different. Processing can yield the different mRNAs. So this processing explains the production of the secreted or the membrane bound forms of the particular immunoglobulin and the simultaneous expression of the IgM and IgD by a single B cell. Okay, so the processing of the mRNA transcript that gives the different immunoglobulins, whether it is IgM or IgD in the B cell. Okay, we'll see how it is. So the expression of the membrane or secreted immunoglobulin, how the membrane or the secreted immunoglobulins are expressed. So we'll see here. So difference depends on the difference of immunoglobulins, that depends on the differential processing of a common primary transcript. So both for the membrane and the secreted immunoglobulin, common mRNA transcript will be present, primary transcript will be present. So the mature naive B cells, that means they are not in contact with the antigen, still they are known as the naive B cells. They produce only the membrane bound antibody. So the mature naive B cells produces only the membrane bound antibody, and differentiated plasma cells produce the secreted antibodies. Okay, there is a difference. So naive B cells produces a membrane bound antibody, and the differentiated plasma cells produce the secreted antibodies. So the secreted form has a hydrophilic sequence of about 20 amino acids in the C-terminal domain. This is replaced by a sequence of about 40 amino acids containing the hydrophilic segment. This extends outside the cell, hydrophilic segment outside the cell, and hydrophobic transmembrane segment that is in the transmembrane, and a short hydrophilic segment at the C-terminal that extends into the cytoplasm. The secreted one and the membrane bound one. Secreted one has the hydrophilic sequence of about 20 amino acids in the C-terminal region, and in case of the membrane bound, sequence of about 40 amino acids that contains hydrophilic segment that extends outside the cell, and hydrophobic transmembrane segment in the transmembrane, and short hydrophilic segment at the C-terminal region. Okay, so this we'll see. So this is the secreted immunoglobulin, and this is the membrane bound. So the secreted immunoglobulin consists of what is known as approximately 20 amino acids hydrophilic sequence at the C-terminal region. So this is the one hydrophilic sequences. So this purple colored one are hydrophilic, and the hydrophobics are this one. Okay, so next one, what is that? In the membrane bound immunoglobulin, so hydrophilic sequence is outside, and the transmembrane hydrophobic, and the cytoplasm again hydrophilic sequences. So this is just for your understanding. Okay, so the next one is how this differential processing of the mRNA transcript takes place. Differential processing. Okay, simultaneous expression of the IgM and IgD. So the controlled by differential RNA processing, if the heavy chain transcript is cleaved and polyadenylated at site 2 after the mu exons, then the mRNA will encode the membrane form of the mu heavy chains. If the polyadenylation is instead further downstream at the site 4 after the C delta exons, then RNA splicing will remove the intervening sequences that produce the mRNA encoding the membrane form of the heavy chains. We'll see here. So this is the primary heavy chain transcript. Primary heavy chain transcript. So poly A adenylation at site 1. This is at the site 2, and this is site 3 and site 4. Okay. So now this is the C mu region, expression of IgM and IgD till here, and from here to here for D, constant region of the D. So polyadenylation at site 1. So if that takes place at site 1, okay. So recombination RNA transcript for the secreted mu. So VDJ, J region, mu1, mu2, mu3 and mu4. So polyadenylation takes place at this region. Okay. So then RNA splicing takes place, the introns are removed, and then you will end up with a mRNA transcript which is coding for mu, that is IgM. So another method. So site 2 means this one. Okay, polyadenylation at this region M2. Okay, so site 2, what happens? RNA transcript for the membrane mu, so then mu1, mu2. So what happens finally? So joining of this, splicing, and finally mRNA encoding for the membrane mu chain. So this is mRNA encoding for the membrane. mRNA encoding for the mRNA encoding for the secreted mu chain and mRNA encoded for the membrane mu chain. So difference in the secreted form of immunoglobulin and the membrane bound form of the immunoglobulin production during RNA processing. So this is how we will end up with the what is known as the primary transcript processing leads to whether it is a membrane bound or the secreted form of the immunoglobulin. Next we'll see simultaneous okay, I think I confused. Okay, this is the membrane or secreted immunoglobulin. So next one is simultaneous expression of the IgM and IgD. So this one, so how this takes place? So this is also almost similar. So till here, polyadenylation of the primary transcript till site 2. So that expresses the what is known as joining, splicing, and we will end up with a mu mRNA. And then this one, polyadenylation of the primary transcript of the site 4 till here. Okay, and this will be eliminated. So in between mu will be eliminated. This part will be eliminated. In the first one, this part will be eliminated. In the second one, during de-expression, this part will be eliminated during splicing, and we will end up with the mRNA that will be transported to nucleus, and it is expressed, translated to particular heavy chain. So this is how the IgD and IgM are expressed. Next, synthesis, assembly and secretion of the immunoglobulins. So this is for the transmembrane. So transmembrane segment, they are present in the secretory vesicle, the fusion with the membrane. So it is targeted to membrane. So this is how the transmembrane or membrane-bound immunoglobulins are formed. So next one, this secreted immunoglobulins. So these are synthesized in the rough endoplasmic reticulum with the leading sequence which is known as, so leading sequence is excised or removed. 1 minute. Leading sequence is excised or removed, and it is processed and transported as a vesicle, and it reaches the cis Golgi complex. So Golgi, transgolgi reticulum, where the glycosylation takes place. Okay, glycosylation takes place in the end of what is that? Golgi complex, and then it forms as a secretory vesicle. Oligosaccharides, along with the oligosaccharides, they are exocytosis. Okay, secreted immunoglobulins are formed. So membrane bound immunoglobulins and the secreted immunoglobulins formation. So next, regulation of the immunoglobulin gene transcription, how that process is regulated. So there are three regulatory sequences in the DNA which regulate the transcription of the immunoglobulin genes: promoters, enhancers, and the silencers. So these three controls or regulate the immunoglobulin gene expression. So promoters, which promotes the initiation of the RNA transcription in a specific direction. Okay, this is located about 200 base pair upstream from the transcription initiation site. Enhancers. So these are the sequences located some distance upstream or downstream from a gene that activate the transcription from the promoter sequence in an orientation independent manner. Silencers, these are located, uh, these are also nucleotide sequences that downregulate the transcription, operating in both the directions over a distance. So each VH and VL gene segment has a promoter located just upstream from the leader sequence. So these promoters contain a data box which serves as the site for the binding of number of proteins that initiates the transcription. You can see here where promoters, enhancers, and silencers are responsible. So promoters are red in color, enhancers are green in color, and the silencers are yellow in color. So these are the promoters, and these are the enhancers, and these are the silencers. So they are present in the AB chain also, K chain also, and the lambda chain also. In all the cases, they are present. So these regulates the transcription. What is that? Transcription of immunoglobulins. So next, effect of DNA rearrangement on the transcription. Effect of DNA rearrangement. So whether the rearrangement is required or not. So what if it is not rearranged? So rate of transcription of a rearranged VL, JL or VH, DH, JH unit is as much as 10,000x the rate of transcription of the unrearranged VL or the VH. That means, so the transcription of the rearranged genes will be more than the transcription of the unrearranged segments. Next, the genes that promote the cellular proliferation are genes that promote the cellular proliferation or prohibit the cell death. So these can often translocate to the immunoglobulin heavy or the light chain loci. So oncogenes they are known as. So genes that promote the cellular proliferation or prohibit the cell death. So these can often translocate to the immunoglobulin heavy or the light chain loci. So expression of these genes is elevated significantly under the influence of an immunoglobulin enhancers, promoters you have studied, no? So that one. Next, inhibition of immunoglobulin gene expression in the T cells. So T cell receptors are given in the syllabus. The T cell receptors are TCR. I've already explained the structure of TCR and all. So in TCR also, that also undergoes the rearrangement, VDJ rearrangement to generate the functional TCR genes. So this occurs by the similar recombination mechanism of Ig, including the recombinases RAG1 and RAG2, similar to immunoglobulin genes. So the complete Ig gene rearrangement of heavy and the light chains occurs only in the B cells. Okay, so immunoglobulin gene, because this is the one which secretes the immunoglobulin subtypes, immunoglobulins, antibodies, okay, in B cells. And the complete TCR gene rearrangement, is limited to T cells only. TCR gene rearrangement is limited to T cells, and the complete Ig gene rearrangement for H and L chains which occurs in the B cells. So this is about the immunoglobulin gene expression. So summary of this, immunoglobulin light and heavy chains are encoded by 3 separate multi-gene families. So functional light and heavy chains are generated by random rearrangement of the variable region gene segments. Next, VDJ joining is catalyzed by the recombinase activating genes, that is RAG1 and the RAG2 and other enzymes and proteins. Joining of segments is directed by the RSS, recombination signal sequences. So Ig gene rearrangements occur in sequential order. So heavy chain rearrangement first, followed by the light chain rearrangement. Next, major sources of antibody diversity are the random joining of the multiple V, J, and D gene segments, junctional flexibility, somatic hypermutation, random association of the heavy and the light chains, P addition and N addition. Okay, so this leads to the antibody diversity. Next, class switching results in the expression of different classes of antibody, whether IgG, IgA, IgE, or membrane bound, are the secreted immunoglobulin. So, with the same antigen specificity. Next, differential RNA processing of Ig heavy chain primary transcript generates the membrane bound antibody in the mature B cells and secreted antibody in the plasma cells, and the simultaneous expression of IgM and the IgD by the mature cells. Okay, class switching that gives the different immunoglobulins, and the differential RNA processing of the heavy chain gives rise to membrane bound or the secreted plasma cells. So simultaneous expression of IgM and IgD takes place by differential RNA processing. Okay, so this is about the immunoglobulin gene expression.